Describe the steps of heme synthesis and its related disorders. Explain with examples the clinical significance of inhibition of heme synthesis and explain how heme synthesis is regulated. Then we will discuss and about the porphyrias, how they are defined and how they are classified, and lastly, list the enzyme deficiencies and key clinical features associated with each porphyria. Before starting this topic, you have to uh, go through this topic from Lippincott. It is divided into two sections. One section is about in the chapter globular proteins, and the other is in the chapter 21, I think, about porphyrias. Okay, as you see this picture, it shows a vampire. What is the link of vampire with this lecture? We will see what is the link. These are the pictures of the person. This is the normal appearance of this boy, and after some time, he develops these facial expressions. And this is also one of the picture of a person who has porphyria. This is the characteristic hallmark of porphyrias, in which the urine of a person after standing turns into brownish reddish colored due to different compounds which are accumulated in the urine. This is also called a vampire disease or porphyrias. It's a rare disease. In 1811, King George III was diagnosed with this, presented with this disease with attacks of severe pain and excited overactivity, paralysis, and delirium. These symptoms began in 1765 at the age of 27, and then they occur frequently. In 1811, he presented as violently insane, and then he turned into blindness. From there, this disease was noticed by the clinicians of that time, and the presenting complaint of King George was he had acute bouts of abdominal pain and mental confusion, may have been a porphyria sufferer or complicated by the drugs that his doctor gave him. This is also called vampire disease or werewolf disease. Some have put forth that porphyrias are misinterpreted, but in Middle Ages, some patient presented with photosensitivity, red blood, or even teeth, hypertrichosis, excess hair growth on the body. So, porphyrins. What are porphyrins? They are basically cyclic compounds that binds metal ions, usually iron, but they can bind any other metal also. Most common porphyrin in human is heme, and one ferrous group in the tetrapyrrole ring, which acts as a prosthetic group. Heme proteins are rapidly synthesized and they are degraded. 6 to 7 grams per day of hemoglobin is turned over. Porphyrins are basically cyclic compounds formed by the linkage of four pyrrole rings. These are the four pyrrole rings. They are joined by methynyl bridges connects these pyrrole rings. They are four, labeled as A, B, C, and D. Okay. They have different groups attached to it. Two groups, acetyl group A and propionyl group B, are found attached to each pyrrole ring on the porphyrin molecule. The nature and distribution of these side chains determines the physical characteristics of porphyrin, that is, they determine the solubility of this compound. Based on the arrangement of A and P groups, the molecules are classified into four groups designated by type 1 to type 4. Type 3 porphyrins contains asymmetric distribution on ring D, and they are basically physiologically important to humans. See in this picture, they in ring A, AP acetyl group, propionyl group. In ring B, acetyl group, propionyl group. Then in C, but in D, their position changes. Propionyl comes first, and acetyl goes in the end. So this change is is basically important for the normal functioning of the porphyrins in the human body, which makes the series of three type 3 series is basically physiologically active porphyrins in our body. The nature and distribution of these side chains, as we have discussed, is determines the solubility of the porphyrin. Porphyrin, Europorphyrin 1 is a compound which has all these four rings attached with methanol bridges, and in this you can see AP, and AP, all symmetrically arranged. Porphyrins contain side chains attached in each of the four pyrrole rings in type 1 porphyrin. These side chains are arranged asymmetrically, that is, Europorphyrin 1 acetate alternates with propionate around the tetrapyrrole ring. But in this Europorphyrin 3, in all three A, B, and C are similar, but in ring D the sequence changes, and this makes this europorphyrin physiologically active in human bodies. So porphyrins are basically cyclic compounds with four pyrrole rings attached by methanol bridges. These side chains may vary. In uroporphyrin, we have acetate and propionate chains, but in corpoporphyrin, we have methyl and propionate chains. An order of the chains defines the subgroup of porphyrins only. Type 3 porphyrins are physiologically important for human body. See this same picture, Europorphyrin 1, all are symmetrically arranged acetyl and propionate groups, but in Europorphyrin 3, the group D has different arrangement. See in corpoporphyrin 1, all A, B, and C D groups are rings are have similar methyl and propionate arrangement. But when in corpoporphyrin 3, when they are changed into corpoporphyrin 3, the sequence changes in group in ring D. We have an important porphyrin 3 or protoporphyrin 3, also called protoporphyrin 9, which is the parent porphyrin of heme, and it, by the action of the enzyme ferrochelatase, converts this protoporphyrin 3 or protoporphyrin 9 by the incorporation of iron into heme, and this heme will form the helps in the formation of synthesis of hemoglobin. Porphyrins act as a tetradentate chelating molecule for metals. The four nitrogen atoms functions as teeth to hold the metal ions in place. These act as teeth to hold the metal ions. Sorry. Chelatase enzyme, that is ferrochelatase, facilitates the insertion of the metal ion into the porphyrin ring. like ferrochelatase, is responsible for insertion of iron into the porphyrin to form heme. After insertion, the metals are held on by electrostatic interactions of the nitrogen electrons. Case study: today's patient, a 6-year-old child brought in OPD with complaints of having learning difficulties in school. On inquiring further, his parents revealed that he has a habit of eating paint chips. What could be the most likely diagnosis? Means, okay, anything that is linking the difficulty in learning at the in his classroom with the eating of paint chips. You think about it, and then at the end of the lecture you will be able to diagnose what is the problem with this child. Heme is basically a derivative of a family of compounds called porphyrins, which we have said, porphyrins are basically cyclic compounds formed by the fusion of four pyrrole rings that are linked by methynyl bridges, and the most important protein contains heme as the prosthetic group. Then there are certain specialized type of proteins that contain heme as a prosthetic group. These are called heme proteins, and they include hemoglobin, whose function is to carry oxygen, myoglobin, which is present in the muscles and causes oxygen transport. Then we have a group called cytochromes, excuse me, cytochromes, which are present in electron transport chain, as well as they are present in the liver and makes the cytochrome P450 enzyme system. Then we have catalase enzyme, that is also a heme protein. So heme structure is, what is a heme structure? Heme structure is a complex of porphyrin 9 and ferrous ion. Porphyrin 9, when incorporates iron in the center with the help of these nitrogen compounds, forms the heme group. Structure of heme is methyl, and in the structure of heme we have methyl and vinyl side chains, in A ring, then methyl and vinyl rings in B ring, then methyl and propional rings uh, in C ring, C pyrrole ring, and methyl and propionyl and methyl ring in uh, group D, ring D. Iron containing porphyrin is a complex of protoporphyrin 9 and iron ferrous iron. The iron is held in the center by the heme molecule by bonds of four nitrogens of the pyrrole rings joined by the four methynyl bridges. Porphyrin is formed by four pyrrole rings. The side chains 1, 3, 5, and 8 are methyl. The side chains 2 and 4 are vinyl, and the side chains 6 and 7 are propionic acid. This makes the heme structure. What is the site of heme synthesis? Basically, in the tissues, heme is synthesized in liver and bone marrow, and at the cellular level, it is synthesized in the mitochondria and the cytoplasm. Biosynthesis of heme. Heme can be synthesized by almost all the tissues in the body, but primarily it is synthesized in the liver and the bone marrow. It is synthesized in the normoblast phase, but not in the mature ones. Because why not in the mature ones? Because in the mature RBC, there are no organelles present. So mature RBC has no organelles. Heme synthesis within the liver. 10% of the heme synthesis in the body is synthesized in the liver, mostly incorporated into the cytochrome P450 enzyme system, which is also responsible for the metabolism of xenobiotics such as drugs and alcohol. Liver heme synthesis is upregulated in response to drugs and alcohol intake or alcohol metabolism. Then we will start the synthesis of heme. There are five. There are certain steps in the synthesis of heme. They are basically divided into 7 steps. Step 1 is formation of delta aminolevulinic acid. Step 2 is formation of porphobilinogen. Step 3 to 5 is the formation of the cytoplasmic tetrapyrrole intermediates. Step 6 is formation of protoporphyrin 9 in the mitochondria. And step 7 is formation of heme in the mitochondria. We will discuss each step in detail. These are the steps of heme synthesis. So, the major site of synthesis in the body is bone marrow and erythroid cells. 85% of the heme produced in the body is synthesized. Its synthesis rate is relatively constant, 10% in the liver and 85% in the bone marrow. Synthesis is upregulated in response to drugs, certain drugs, and alcohol metabolism. The cellular location is mitochondria. Initial reaction and the last 3 steps occur in the mitochondria, while the intermediate 4 steps occurs in the cytosol. Mature RBC don't have mitochondria, so they cannot make heme. So the precursor or the basic uh, molecules which require for the synthesis of heme are succinyl CoA. From where we get succinyl CoA? We get from TCA cycle. Succinyl CoA combines with glycine, which is an amino acid, and by the enzyme of named ALA synthase, because something is going to be synthesized at this step. So ALA synthase will help succinyl CoA joined with glycine to form al- aminolevulinic acid. Once this delta aminolevulinic acid is formed in the mitochondria, it goes into the cytoplasm. Then in the cytoplasm, this ALA or aminolevulinic acid is changed into porphobilinogen by the enzyme dehydratase. Then protoporphyrinogen will be converted into hydroxymethylbilane by the enzyme hydroxymethylbilane synthase. Then uroporphyrinogen 3 is formed by the enzyme uroporphyrinogen 3 synthase. See, see this, okay, new compounds are synthesizing. So the enzyme mostly used are synthase. Then decarboxylation of uroporphyrin 3 occurs by the enzyme decarboxylase and it is changed into corpoporphyrin 3. Once this compound is formed, it enters the mitochondria again and it is then changed into protoporphyrin 9 by the enzyme corpoporphyrin oxidase. Then protoporphyrin 9 is converted into protoporphyrin by the enzyme protoporphyrin oxidase into protoporphyrin 9. And once when iron is incorporated into this compound by the help of the enzyme ferrochelatase, then heme is synthesized. So these are the steps of heme synthesis. Formation. The first step is ALA or delta aminolevulinic acid. This is the most committed or rate limiting step of the porphyrin synthesis, which occurs in the mitochondria, and glycine and succinyl CoA condenses to form ALA, delta aminolevulinic acid. Glycine is a non-essential amino acid, while succinyl CoA is an intermediate of TCA cycle. And this reaction is carried out by the enzyme ALA synthase, and it requires a coenzyme called pyridoxal phosphate, which is necessary for the carrying out of this reaction. This is the first step which occurs in the mitochondria, and this is the rate limiting or committed step of heme synthesis. ALA synthase is a mitochondrial enzyme. It has two isoforms. ALA S1 is located mostly in the liver cells and to a smaller extent in other cells of the body as well. Heme production by the liver cytochrome P450 monooxygenase system is involved in this reaction at the level of the liver cells. This its gene is located on chromosome 3, and loss of function in the mutation in LRS1 gene causes impaired metabolism of xenobiotics. LRS2 is produced in erythroid tissues and heme production for RBC hemoglobin. Its gene is located on X chromosome. Loss of function or mutation in LRS2 results in anemia. So, porphyrin biosynthesis is primarily regulated by LRS regulation. It's the regulatory enzyme. Again, now stepwise we will see in the step 1, which occurs in the mitochondria, an intermediate of TCA cycle, succinyl CoA, incorporates with a non-essential amino acid glycine, and with the help of the enzyme ALA synthase and pyridoxal phosphate as a coenzyme. And this condensation reaction condenses to form the delta aminolevulinic acid. This is the committed rate limiting step of heme synthesis. You have to remember this step. Then, once ALA is synthesized, it enters the cytosol, and two condensation of two molecules of delta aminolevulinic acid will form protoporphyrinogen under the with the help of the enzyme ALA dehydratase. This is a zinc containing enzyme and is sensitive to inhibition by lead. This is the first protoporphobilinogen PBG is the first precursor of pyrrole, which is formed in the cytosol at step 2 level. Then step 3 also occurs in the cytosol, and four molecules of porphobilinogen condenses to form hydroxymethylbilane by the help of the enzyme uroporphyrinogen 1 synthase. This forms the linear tetrapyrrole chain. Then at step 4, hydroxymethylbilane will be changed into will be changed which can be changed spontaneously into uroporphyrinogen 1 or by the enzyme of uroporphyrin 3 synthase, it can be changed into uroporphyrin 3. This forms the norm. This is formed under normal condition. Then step 5 occurs in the mitochondria, and uroporphyrin 3 by the enzyme uroporphyrin decarboxylase is changed into corpoporphyrin 3, and corpoporphyrin 3 then changes into protoporphyrin 9 by the enzyme corpoporphyrin oxidase. This step occurs in the mitochondria. Then protoporphyrin 9 incorporates iron by the enzyme by protoporphyrin oxidase and will form protoporphyrin. Then at step 8 in the mitochondria, protoporphyrin 9 incorporates ferrous iron by the enzyme ferrochelatase will form the heme. Formation of tetrapyrrole intermediates. Condensation of 4 porphobilinogen produces the linear tetrapyrrole molecule, that is hydroxymethylbilane, catalyzed by the enzyme hydroxymethylbilane synthase. Cyclization and isomerization of hydroxymethylbilane to uroporphyrin 3 forms the tetrapyrrole ring, catalyzed by uroporphyrin 3 synthase. Decarboxylation of acetate groups of uroporphyrin 3 to generate corpoporphyrin 3 is catalyzed by uroporphyrin 3 decarboxylase, and these reactions all occur in the cytosol of the cell. Corpoporphyrin 3 enters the mitochondria, and two propionate side chains are decarboxylated to vinyl groups, generating the protoporphyrin 9. Oxidation of protoporphyrin 9 to protoporphyrin 9. The introduction of iron as ferrous into the protoporphyrin 9 to form the heme. Last step in the formation is the incorporation of iron into the porphyrin ring. The introduction of iron into the porphyrin 9 occurs spontaneously by the electrostatic interactions. The rate of iron incorporates is enhanced by the enzyme ferrochelatase. Ferrochelatase is inhibited by lead. The other enzymes in porphyrin biosynthesis that is inhibited by lead is ALA dehydratase, as we have discussed in the second step. So, lead poisoning. How lead poisoning occurs? Lead, as you see in this meta formation of heme, at two steps, at this step number 2 and step number 7. The lead acts as a cofactor in these two steps. And whenever a person is consuming lead either through eating of paints or drinking heavily or exposure to uh, smoke or the things that are high in lead, then this lead will halt this formation of heme at two steps. Ferrochelatase and aldehydatase are inhibited by the lead. So protoporphyrin and ALA accumulate in the urine in lead poisoning. Lead poisoning occurs when these two enzymes aldehydatase and ferrochelatase are inhibited. Other heavy metals may also inhibit these enzymes, and symptoms resembles porphyrias. ALA and protoporphyrin then are excreted in the urine. How lead poisoning occurs? Lead poisoning is basically occur by lead pipes, by paints, by smoke, and by uh, pollution. Symptoms occurs in lead poisoning are usually headache, irritability, reduced sensation, aggressive behavior, difficulty sleeping, abdominal pain, poor appetite, constipation, and anemia. Additional complication for children can be loss of developmental skills, problems in behavior or attention, hearing loss, kidney damage, reduced IQ, and slowed body growth. These all can lead to lead poisoning and thus can lead to anemia in children. This is the overview of heme synthesis. Succinyl CoA with glycine forms the delta aminolevulinic acid by the enzyme ALA synthase. Then delta aminolevulinic acid leaves the mitochondria and goes in the cytosol where it is converted into porphobilinogen, then successively into uroporphyrinogen, then corpoporphyrinogen 3. Once corpoporphyrin 3 is formed, it enters the cytoplasm. From the cytoplasm, enters the mitochondrial matrix again, and then it is converted into protoporphyrin 9, then protoporphyrin, and then heme. Now, we will summarize this heme synthesis again. Succinyl CoA joins with glycine and form delta-aminolevulinic acid by the enzyme delta-aminolevulinic acid synthase, which this enzyme requires pyridoxal phosphate as a coenzyme, and this heme can inhibit this step. Thus, this is the most committed or rate limiting step of heme synthesis. Once delta-aminolevulinic acid is formed, it is changed into porphobilinogen by the enzyme delta-aminolevulinic acid dehydratase. If this step is uh, not carried out properly, either due to lead poisoning or due to the deficiency of this enzyme, then a porphyria occurs called delta aminolevulinic acid dehydratase porphyria. From here now on, porphobilinogen is formed, then hydroxybillane, then uroporphyrin 3, then corpoporphyrin 3, then protoporphyrin 9, and protoporphyrin 9, and then once iron is incorporated by the enzyme ferrochelatase, then heme is synthesized. These are the different types of porphyrias which occur at the level of specific enzyme deficiencies. If porphobilinogen deaminase is absent, then acute intermittent porphyria can occur. If uroporphyrin 3 co-synthase is absent, then congenital erythropoietic porphyria can occur. If uroporphyrin 3 decarboxylase is absent, then porphyria cutanea tarda can occur. If corpoporphyrin oxidase is absent, then hereditary corpoporphyria will occur. If protoporphyrin oxidase is absent, then variegate porphyria may occur. And if ferrochelatase is absent, then erythropoietic protoporphyria will occur. This heme synthesis, if any of these compounds are accumulated due to the deficiency of their specific enzyme, then this heme synthesis cycle will be stopped. Accumulation of heme gives a negative feedback response to Ella synthase to inhibit this new formation of heme. These are the similar which we have discussed. These are the enzymes which are deficient and with their specific disease occurs with the deficiency of specific enzymes. If delta 1 levulinic acid synthase is deficient or absent, then sideroblastic X-linked anemia can occur. Similar is this porphyria, different types of porphyrias. How is LAS1 is regulated? LAS1 is mostly found in the liver, and heme is made from liver cytochrome P450 monooxygenase system, that is heme containing enzymes. Cytochrome P450 is involved in the metabolism of xenobiotics. Heme utilizes led to lower cellular heme utilization leads to lower cellular heme concentration, and lower heme concentration needs for heme leads to increased synthesis of LAS1. Excess of heme, on the other hand, inhibits LAS1 production. How is LAS2 regulated? It is regulated post transcriptionally by cellular iron status. LAS2 messenger RNA has a iron response element sequence in the 5' untranslated region. Iron response regulatory protein can bind to iron response element, and this binding blocks the messenger RNA translation and production of LAS2 protein. Then LAS2 protein production is inhibited, and when iron levels in the cell is low, heme is not available for hemoglobin synthesis. In erythroid cells, heme and globin synthesis need to be coordinated to meet the needs of developing RBCs. In the absence of heme, globin synthesis is downregulated. LAS1 is inhibited by excess of heme. When excess heme is accumulated, this gives a feedback repression to LAS1. It can also be inhibited by three other mechanisms. One is called hemin, that is, oxidized heme inhibits the transcription of LAS1 gene. Free heme interferes with the LAS1 messenger RNA stability and inhibits its translation. Free heme can also interfere with translocation of the LAS1 enzyme into the mitochondria, and this is how LAS1 regulates heme synthesis. Regulatory mechanism of heme synthesis is done by ALA synthase, which controls heme biosynthesis in the liver, that is the committed rate limiting step. In liver, ALA synthase activity is controlled by feedback inhibition of heme, which is the last step of heme synthesis. And when heme accumulates, then it causes feedback inhibition of ALA synthase. Then there is also a phenomena called repression. Heme acts as a co-repressor and combine with aporepressor to form a holorepressor, and thus prevents synthase gene. Derepression means induction. This occurs in the presence of certain drugs like barbiturates or steroids due to diversion or increased utilization of heme by cytochrome P450 hydroxylase system. RBC maturation cannot occur, and heme synthesis stops as RBC maturation occurs. Regulation of heme synthesis. Synthase is regulated by repression mechanism. Heme inhibits the synthesis of synthase by acting as a co-repressor. ALA synthase is also allosterically inhibited by hemetin. When there is excess of free heme, the iron is oxidized to ferrous iron is oxidized to ferric iron, thus forming hemetin. Disorders of heme synthesis. Porphyrias are a group of inborn errors of metabolism associated with the biosynthesis of heme. Porphyria comes from a Greek word meaning purple. They are characterized by increased production and excretion of porphyrin compounds or their precursors like ALA or PBG. Many porphyrias are inherited as an autosomal dominant trait. A 5-year old golf instructor presented with painful blisters on the back of his hands. He had partial complex seizure disorder that had begun about 3 years earlier after a head injury and had been taking phenytoin since the onset of his seizure disorders. He takes an average weekly ethanol intake of about 18 to 12 ounce cans of beer. The patient's urine was reddish orange. Cultures obtained from skin lesions failed to grow any organism, so a 24-hour urine collection was done that showed elevated levels of uroporphyrin. Normal level is less should be less than 27 milligram per dl, and elevated uroporphyrin was found of 1000 milligrams per dl. What could be the likely reason in this case? As you see, this is a 50-year-old golf instructor. He spent most of his time in the sun, and under the exposure of sun, he develops painful blisters. He is also having a history of seizure, and for this, he has been taking phenytoin for a long time. He is also a alcohol. Uh, uh, he also takes alcohol in the form of beer. This patient develops this problem. His urine turned reddish orange in color after standing for some time, and this patient has been diagnosed as a case of porphyria cutanea tarda. Porphyrias are genetic diseases in which activity of one of the enzymes involved in heme synthesis is decreased. Symptoms vary depending on the severity of the deficiency, whether heme synthesis is affected primarily in the liver or in the erythrocytes. Common symptoms of porphyria are abnormalities of the enzyme of heme synthesis, accumulation of ALA or BPG or decrease in heme synthesis in the cell or body fluids causes neuropsychiatric signs and symptoms. But when porphyrinogens are accumulated in the skin or in the tissues, then spontaneous oxidation leads to photosensitivity. So, a person of porphyria can present with two basic problems: neuropsychiatric signs symptoms or with photosensitivity, depending on the enzyme deficiency. Common symptoms of porphyria. Occasional episodes of severe neurological symptoms are associated with some porphyrias. Had acute bouts of abdominal pain and mental confusion. This is usually the common presentation seen of this disease. Permanent nerve damage and even death can result if it is not treated promptly. Elevated ALA arising from derepression of ALA synthase gene transcription is considered responsible for neurological symptoms. When there is accumulation of ALA, then the patient usually presented with neurological symptoms. Porphyrias are broadly grouped into three types: hepatic porphyrias, erythropoietic porphyrias, or porphyrias with both erythropoietic and hepatic abnormalities. They can also be classified as hereditary or acquired. Hereditary are autosomal dominant or autosomal recessive diseases like congenital erythropoietic porphyria, while acquired are due to lead poisoning. A, porphyrias can also be classified as hepatic or erythropoietic. Hepatic can be of acute origin or of chronic origin. Acute porphyrias are acute intermittent porphyria, variegate porphyria, hereditary corporoporphyria, and delta aminolevulinic acid dehydratase deficiency porphyria. Non-acute porphyrias are porphyria cutanea tarda, erythropoietic protoporphyria, and congenital erythropoietic porphyria. Porphyrias result from defect in heme biosynthesis pathway. Genetic disorders mostly occur due to mutation leading to the deficiency of an enzyme in the heme biosynthetic pathway. Complete absence of a heme pathway enzyme is incompatible with life. So, accumulation and increased excretion of porphyrins or porphyrin precursors in patients is usually the common presentation. Urine color is altered in some patients. In some cases, it can turn purple or reddish orange in color, and hence, due to this change in color, it is called porphyria or purple color disease. Classification of porphyrias, they are based on the affected step in the heme biosynthesis pathway. Enzyme defect prior to the synthesis of tetrapyrols, yani before the synthesis of tetrapyrrole rings occurs. Affected individuals manifest with abdominal pain and neuropsychiatric symptoms or signs. Enzyme defects leading to accumulation of tetrapyrrole intermediates are usually presented with photosensitivity, and exposure to skin by sun rays causes severe itching and burning of the skin. Light exposure causes tetrapyrrole photooxidation, reactive oxygen species formation, and thus causing oxidative damage to the skin cells. Based on body cells and tissues where heme biosynthesis is affected, erythropoietic porphyrias, heme biosynthesis in the erythropoietic cells of the bone marrow is affected, while in hepatic porphyrias, heme synthesis in the liver is affected. Acute hepatic porphyrias occur due to deficiencies of the dehydratase and ALA accumulate, and thus ALA accumulates in the urine. Tetrapyrrole intermediates do not accumulate, and there is no sensitivity. Mostly in this case, they have neuropsychiatric problems. Hydroxybilane synthase deficiency will occur at this point. ALA and BPG both accumulate in the urine, and they causes the urine to change its color on exposure to air. Tetrapyrrole intermediates do not accumulate, and no photosensitivity is seen up till this step of deficiency. The first tetrapyrrole structure to be formed is hydroxymethylbilane, which is a linear tetrapyrrole. Then acute hepatic porphyrias. ALA dehydratase deficiency and hydroxymethylbilane synthase deficiency leads to severe abdominal pain, neuropsychiatric disturbances ranging from anxiety to delirium, symptoms precipitated by intake of certain drugs like barbiturates, ethanol, which induces the synthesis of cytochrome P450 enzyme system. This further decreases the amount of available heme, which in turn promotes the increased synthesis of ALA-S1. Then we have chronic hepatic porphyrias. Accumulation of hydroxymethylbilane leads to spontaneous non-enzymatic formation of uroporphyrin 1 and corpoporphyrin 1. Uroporphyrin 1 and corpoporphyrin 1 undergoes oxidation and are elevated in the urine, and therefore uh, urine is therefore pink to red under ultraviolet light. Occurrence of porphyria cutanea tarda is further aggravated by factors like hepatic iron overload, alcohol abuse, hepatitis B or C, or HIV infections, or light exposure causing high photosensitivity. In erythropoietic porphyria, heme biosynthesis in the erythroid cells of the bone marrow is affected. It is of two types: congenital erythropoietic porphyria, in which uroporphyrin 3 synthase is deficient, while erythropoietic porphyria, in which there is deficiency of the enzyme ferrochelatase. In this, from here, the patient usually presented with dark color or pink or reddish color urine, and patient have photosensitivities. Ferrochelatase deficiency can occur and patient presented with photosensitivity. Por- Porphyria cutanea tarda is a chronic porphyria and is the most common type. Liver and erythroid tissues are affected, and there is deficiency of uroporphyrin decarboxylase. Often, no symptoms occur until the fourth or fifth decade of life. Clinical expression is determined by many factors. Hepatic iron overload can occur, exposure to sunlight can be a factor, hepatitis B or C or HIV can be aggravating factors. Symptoms usually presented with cutaneous rashes or blisters. Urine is red to brown in natural light or pink to red in ultraviolet light. Chronic hepatic porphyria or porphyria cutanea tarda, caused by the deficiency of the enzyme uroporphyrin decarboxylase, occurs late onset in the fourth and fifth decade of life, and accumulation of this compound leads to red to brown colored urine and skin eruption. This is the presentation, usual presentation of porphyria cutanea tarda patient. Uroporphyrin accumulates in the urine, and clinical features are cutaneous photosensitivity, and liver exhibits fluorescein due to high concentration of accumulated porphyrins. This is the picture of your book, which shows different types of porphyrias with their deficiency states and the intermediate accumulation with sign and symptoms. Porphyrins are obligate intermediates on heme synthetic pathways. The first step, ALA, is formed, then PBG, then uroporphyrin, then C7 porphyrin, then C6 porphyrin, then C5 porphyrin, then corpoporphyrin, then protoporphyrin, then protoporphyrin, and then heme. Each step is catalyzed by an enzyme. These are the enzymes which we have discussed, and these are the deficiencies deficiency diseases that can occur. A deficiency of these enzymes can lead to different types of porphyrias. You have to go through each and every step, especially learn the rate limiting step and the coenzyme or cofactor which are regulating these steps, and the specific porphyria which is occurring at the deficiency of